Hello, everyone, and welcome to our webinar, Antibody Responses in COVID-19, Implications for Understanding Antibody Testing. Today, our speaker is Tom Fabian. Tom is a clinical education consultant and science advisor for Diagnostic Solutions Laboratory. He is a former biomedical research scientist with deep expertise in the role of the human microbiome in health, chronic disease, and aging. Tom's primary focus is on providing educational resources and consulting services for integrative and functional medicine practitioners, as well as consulting and advisory services for clinical testing laboratories. As a functional nutrition practitioner, he also works with clients on a limited basis on improving gastrointestinal health and optimizing healthspan. So Tom, whenever you're ready.
Thanks so much, Allie. It's great to be here today. So the topic that we're going to be talking about is on antibody responses in COVID-19, and especially focusing on the implications for how do we use that information to understand antibody testing. So of course, there's a lot of information out there now about antibody testing. These types of tests have been out there for several months. There's a lot of conflicting information, especially in the media, in terms of their effectiveness and lots of other features of antibody testing. So what I hope to do today with this information that I'm presenting is clear the air based on what's known from research to date. So again, it's still pretty early in the process. There's still a lot more research that needs to be done to really fully understand antibody responses in COVID-19. But this is really going to give you a good summary of what we know to date. And then that can help you better understand and interpret the antibody testing. All right, so let's start off with the outline. So in terms of the topics we're going to cover today, so I just want to touch a little bit in the beginning on letting you know about the antibody testing option that we offer here at Diagnostic Solutions Lab, and then also a little bit of the additional information that we provide about this test. Mostly what we're going to focus on today, though, is covering these other bullet points. A few of the basics on the actual virus itself that causes COVID-19, that's SARS-CoV-2, as well as some antibody and immunity basics, and then covering some of the key research studies that have been done to date that help us better understand certain aspects of antibody responses to COVID-19 or in COVID-19, so that clinicians, again, can better understand what to expect, what's the best timing for testing, how to understand different responses in terms of patients that have significant symptoms versus asymptomatic, et cetera. And then we'll talk a little bit about seroprevalence. And then lastly, we'll touch on another aspect of immunity, which is memory T-cell responses. All right, so let's go ahead and dive into the first part and just talk a little bit about the testing option that we offer. So we actually have three different testing options for SARS-CoV-2. So we have a diagnostic test, that's a nasopharyngeal swab test that uses RT-PCR, and that's a diagnostic test for COVID-19. We also offer a stool test that's also looking for evidence using RT-PCR of the virus in stool samples. So mostly today we're going to be talking about, of course, antibody testing. We do offer an antibody test that covers both IgG and IgM responses. Again, this is in COVID-19. So I want to talk a little bit more about that so that you understand, again, the context for interpretation. You can get more information about our testing on our website. So if you go to the COVID antibody testing section, you'll see that there's a sidebar for articles on our tests, and there's one in particular that covers the technical details related to our test. So this article is titled Novel Coronavirus COVID-19 IgG and IgM ELISA Tests Performed by Diagnostic Solutions Lab. And in that resource, which is a downloadable PDF, we have all the information related to various things here, and I'll cover that in a second here. So that's an article that goes over the kit methodology, analytical performance, and specifically gets into details of the limit of detection, as well as the coefficient of variation, repeatability, reproducibility, as well as assay cross-reactivity. So that all has to do with the technical aspects of the performance of the test. And then there's also clinical sensitivity and specificity. There's data provided by the manufacturer. So this is a kit that's provided by a company called Epitope Diagnostics. And then there are also third-party studies done by various research groups, hospitals, et cetera, that have been published that also report on the performance of our test and other tests. So you have a very comprehensive documentation here of the performance of the test that we're using and that we're offering, along with a reference list. So I just want to back up here to this particular review. So the title is COVID-19 Serological Tests, How Well Do They Actually Perform? So of course, this is getting into that topic that's very controversial in terms of all this conflicting information on the value of antibody testing, how well do antibody tests perform. So they have a summary statement here that I wanted to just quickly read, which is serological diagnostic tests are being increasingly used to provide a broader understanding of COVID-19 incidence and to assess immunity status in the population. However, there are discrepancies between the claimed and actual performance data for serological diagnostic tests on the market. The findings of this review highlight the need for ongoing independent evaluations of commercialized COVID-19 diagnostic tests. So again, that's why we provide these resources on our website to help you understand that information that you need to have at hand in order to evaluate this test from the performance standpoints. And again, this is just an example of some of the information that's in that document. This is a list of published studies to date done by third parties using this particular kit. Okay, so that's just a little bit about the antibody test that we offer. So now I want to dive into some of the basics around SARS-CoV-2, as well as antibody testing and immunity basics. So this is a schematic of the SARS-CoV-2 virus. So it's showing you the viral structure, some of the key aspects of the structure. And this is important to know for understanding how antibody tests work, how immune responses work to the various antigens on the virus. So we know that the main antigens that the immune system often reacts to are noted here. So you'll see on the right, there's a key for the designation. So there's the spike protein, which is the green structure. So that's the structure that the virus uses to attach to human cells. We'll get into that in a bit more here in a bit more detail. But basically, these key structures are the spike protein, envelope, membrane, and nucleocapsid. The first three are outer structures that are part of the membrane. So those are outer structures that antibodies can fairly easily recognize. But there's also the nucleocapsid, which is an internal protein that has several functions, but it also helps to essentially kind of provide structure to the RNA genetic material. And that's actually one of the key immune generating proteins as well. So this is a schematic showing kind of in a simplified way the overall humoral immune response against SARS-CoV-2. So you can see the virus on the left, and again, it's indicating some of the key structural proteins that antibodies are generally produced in response to. And then there's a zoom in here so you can see basically how this spike protein interacts with a particular receptor in the membrane of host cells, and in particular lung cells. So there's the expression of the ACE2 receptor, and that is the key protein that the spike protein of the virus interacts with that generates the whole process of infection. So that sort of initiates the first step where the virus is then taken up into the host cell, which you see here at the bottom left. In the host cell, then the virus replicates. So it sort of co-opts the genetic machinery, the translational machinery in the host cell to produce and replicate more viruses. Then the viruses are released, and that's where they start to elicit immune responses. So the first immune response is always the innate immune responses. So that's cells like macrophages, dendritic cells that are generally known as antigen-presenting cells. So you'll see that here in the bottom right, this sort of oddly shaped cell represents dendritic cell or macrophage. And again, that's referred to as an antigen-presenting cell. Those types of cells take up the virus, and then they basically process the various antigens and then present them to the adaptive aspects of the immune response, so the T cells in particular. And so basically through that interaction of these antigen-presenting cells with the T cells, that starts a cascade of immune responses in process. So that basically causes the T cell to be activated, and then that helps to stimulate and activate B cells at the same time. And of course, B cells are the antibody producing cells. T cells have compatible or parallel roles in immune responses as well. And there's a certain type of T cell called a cytotoxic T cell that helps identify and then destroy infected cells that are infected with the virus. So you can see here later in the process, kind of in the upper right, that these B cells, once they're activated, become plasma cells. So they're replicating, then they differentiate, and their goal is to produce large amounts of antibodies against the antigens of the virus. So that's what the secrete antibodies are for, and that's where they come from. And you'll note here in the center that one of the key steps in this whole immunity process is once these antibodies are produced, especially to the ones that are targeted to the spike protein, that process, if it inhibits the interaction of that virus with other cells and prevents infection, then that's called neutralization. Then there's other types of B cells that become memory B cells, and that's important for long-term immunity. So this is a great schematic summarizing the humoral immune response to SARS-CoV-2. So it illustrates some really important concepts that help understand the process. This is just the reference for that figure. This is just kind of honing in on the concept of neutralization. So this is really illustrating that a little bit more clearly. So you can see here on the right, on the left, that without antibodies interfering with the binding, then the spike protein can bind directly to the ACE2 receptor. But once neutralizing antibodies are produced, and sometimes that can take a bit of time, and that can be a variable process, but when neutralizing antibodies are produced, those are targeted to the spike protein, and because they're bound to the spike protein, they prevent interaction with the receptor. And so again, that's called neutralization. So this is another schematic illustrating the role of memory B cells and long-lived plasma cells in immune memory. So again, you can see here illustrated on the left that there's this concept of the innate immune cell, the antigen presenting cell, interacting with a T cell, and then influencing the functions of an initially naive B cell. So this is in the early phases of acute infection. And then these B cells can differentiate into short-lived plasma cells. So they can develop into short-lived plasma cells that generate antibodies. And that's important for this initial antibody response that starts to build up after a number of days after infection begins. And then for long-term immunity, those can become long-lived plasma cells that continue to produce antibodies, very specific, very targeted antibodies to that virus long-term. And in some responses, these types of plasma cells can live for many decades. So these long-lived plasma cells are really important for maintaining long-term antibodies in serum. Then B cells can also develop into memory cells, and again, those can persist long-term as well. And they're basically poised to be activated whenever the antigen is encountered again in the future. So they're important more for kind of mounting additional antibody and other responses. But as far as the persistence of antibodies themselves in serum, that's largely due to these long-lived plasma cells. So again, long-lived plasma cells and memory B cells form an important part of long-term immunity. Okay, so that's just a little bit about the virus itself, its structure, and then immunity basics. So let's go ahead and talk about what some of these key research studies that have been coming out are revealing about the details of these antibody responses especially when it comes to time course. So how long does it take the different types of antibodies to develop? Do they tend to stay elevated for a certain period of time, or do they tend to come down? What levels of antibodies are generated based on different levels of disease severity, for example, and also what's the overall seroprevalence in the population. So these are the kinds of things that are being addressed by these studies. So this is kind of a simplified summary diagram based on some of these initial studies as far as the time course for detection of the viral RNA, as well as detection of two of the key antibodies, IgG and IgM, to the virus itself. So you can see here, based on this diagram, and the reference here is in the upper right, that the y-axis represents sort of just in general detection levels versus time in days on the x-axis. And then below that, you can see indication of the results for molecular testing. And again, this is just sort of an overall summary of what's typically seen. There's lots of individual variation, which we'll see in a moment. But in general, you can see that when it comes to molecular tests, because the viral titers tend to be highest in the first few days after onset of symptoms, that's sort of the window for the highest level of positivity of diagnostic tests, so these PCR-based tests. When it comes to the antibodies, however, they take a bit longer to develop. And so typically IgM is the first type of antibody to develop, so it's a bit more reflective of an active infection, although it can persist past the period where the virus is detected. So just be aware that is one of the early antibodies, but doesn't necessarily completely overlap with the viral infection itself. Generally, though, over time, the IgM levels tend to peak within 2 to 4 weeks, and then they start to drop off. And in some studies, they've shown that the IgM levels are no longer detected in many individuals past, say, 4 to 6 weeks. And then the IgM is more indicative of sort of the long-term immunity. And that one is typically something that develops a bit later, although that's, again, pretty variable from person to person. Some studies indicate that in some individuals, the IgG can be detected before IgM. So again, there is some variability, so this is just sort of an average representation. Then the IgG antibodies tend to peak at some point with acute infection, and they generally do start to decline in most viral infections up to a point, but then a certain percentage of them tend to persist when there's long-term immunity. So you can see here that there's kind of an indication of when the molecular test is potentially positive, meaning the diagnostic test looking for the virus, versus the serology test, which is the antibody test, which can start to be positive in some individuals after a week, but generally doesn't become positive in the majority of people that generate immune responses until after 2 to 3 weeks. This is a similar diagram with a little bit more information from a different reference, which I'll show momentarily. But again, you can see here, this shows a little bit more clearly where they're emphasizing at the bottom the window of false negatives. So when it comes to the RT-PCR test, which is shown here in the left, bottom left, and that is indicated by viral load in the curve. So that's the blue curve. So viral load starts to increase early on, even before symptoms start to appear. Then when symptoms appear, that's where the 0 is indicating. They tend to increase over a certain period of time, and of course decrease along with symptoms. And again, we're showing here illustrating here the IgM and IgG responses, which is similar to the previous curve. IgM usually appears a little bit before the IgG responses. But then the key thing here is the sensitivity. So that's related to the sensitivity of antibody testing. So there's antibody testing sensitivity that's related to the performance of the test itself, but of course also to the timing and the presence of the actual antibodies in the serum. And so basically that overall level of sensitivity is going to keep increasing during the period where antibodies are starting to appear, and then tends to plateau after 2 to 3 weeks. So that's primarily what this is showing. So the window of false negatives for the RT-PCR test tends to be either too early before there's enough virus to detect, That's what the red is showing. And towards later on in the infection when the viral titer drops off. Versus the antibody test, so the serological test, basically there's going to be a lot of false negatives if the testing is too early. And so essentially, again, after 2 to 3 weeks, then most people that have been infected and have symptoms will be seropositive, meaning that they will have detectable antibody levels. Okay, so that's this is the reference for that particular figure. But that's really kind of a summary of what we're seeing from a variety of studies. But again, there's a lot of individual variation, which we'll see in some of these additional studies here. So this next one I wanted to highlight is titled Novel SARS-CoV-2 Specific Antibody and Neutralization Assays Reveal a Wide Range of Humoral Immune Responses During COVID-19. And in their summary, they say, we found striking differences in total antibody levels and neutralization titers between hospitalized or severe COVID-19 patients relative to outpatient or convalescent plasma donors, which were obtained with the purpose of transfer to treatment of patients. Significant correlations between IgG and neutralization titers, age, duration of disease, and neutralizing antibodies to SARS are also observed. So we'll look at some of that detailed data here. So in this figure, they're looking at the two different charts here, diagrams. On the left, you'll see it says SRBD. So that's basically an IgG antibody test looking at responses to the spike protein. The RBD stands for the receptor binding domain. So that's the particular part of that spike protein that binds to the ACE receptor. So again, this is a slightly different antigen that's used to target in this test versus the nucleocapsid. And just kind of a side note is, once again, the spike protein, especially the receptor binding domain, and the nucleocapsid tend to contain the immunodominant domains that generate the vast majority of immune responses or antibody responses to the virus. So that's why these are often used in these types of antibody assays. Then on the left column, so the y-axis, you'll see antibody levels. And this is order of magnitudes. It's a logarithmic scale. So the thing to note here is from the key in the upper right, this is indicating the different types of patients or groups of patients that they looked at. So there's the healthy controls in green, and those are at the bottom here showing that they had basically virtually undetectable levels on average. And then the plasma donors and the outpatients. So plasma donors presumably were recovered, may have been milder cases, may have been kind of further down the curve where their antibody levels might be slightly lower than when they had the acute infection. Then there's the hospitalized patients, and then the worst patients, the ICU patients as well. So you can see that those are clustered near the top. So this is just showing that there's a wide range of antibody responses in terms of titers or levels based largely on the severity and kind of whether or not they're acute or post infection. So that's kind of the general trend here that you see is for the most part, and this is supported by lots of different studies now, that the more severe the COVID symptoms and the more severe the case, generally the higher the titers of antibodies there will be. And this is showing that same result for both of the different types of antigens. All right, so I'm just going to look at this next one here. So it's a different study. The reference was just in the previous slide. And again, this is kind of just looking at or illustrating the variability in individuals. So you can see here that this is an antibody test looking at IgM antibodies. And this is basically from time 0, so essentially symptom onset, over time in days. And then in this case, they're illustrating curves, longitudinal curves for individual patients. You can see that down below, patient 1, patient 2, et cetera. So you can see that the time course for IgM is pretty variable, although they say the median time to IgM seroconversion, meaning when that's IgM antibodies are detectable, is about 5 days. So that's kind of an important thing to understand is, again, we look at these standard curves and we see that our whatever patient we're working with, if they're tested within that window, but they may be negative, that doesn't necessarily mean that they don't have any antibodies. That may mean in some cases that they're just tested too early, depending on the time. As you can see, some patients here didn't really seroconvert until further along after about 8 to 10 days. So again, a lot of variability, especially when it comes to IgM. IgG was a little bit more consistent, but again, as far as the time course, there was still some significant variability within that first week or so, where some didn't start to increase in terms of their IgG production until after a number of days. And this is just looking at the curves side by side. So kind of the take home overall is that there is individual variability in how quickly they generate antibodies, and especially when it comes to IgM. But the main take home, and this is something that is supported by other studies, is that especially when it comes to IgG, the vast majority of symptomatic patients, as far as we know, tend to seroconvert, meaning that they're positive for antibody tests for IgG, usually by about 14 days. And then some studies indicate it may be more kind of in the 20-day time frame. So basically 2 to 3 weeks is the time frame where most patients, if they're going to generate antibodies, that's usually the time frame where they can be detected. All right, so this is the next study I wanted to focus on. And this is a study that's received a fair amount of attention recently because one of the things that was kind of probably to some extent overemphasized from this study is that antibodies tend to decline over time. So there hadn't been very many studies up to this point looking longer term, just because, of course, the pandemic is fairly new. Not a lot of studies have looked at these things long term yet. So the summary I want to read here is using sequential serum samples collected up to 94 days post onset of symptoms from 65 RT-qPCR confirmed SARS-CoV-2 infected individuals. So these are confirmed positive COVID cases. We show seroconversion in greater than 95% of cases and neutralizing antibody responses when sampled beyond 8 days post onset of symptoms. We demonstrate that the magnitude of this neutralizing antibody response is dependent upon the disease severity, but this does not affect the kinetics of the neutralizing antibody response. So a few key points here, and again, we'll look at the data here in a moment, is number one, that most people that were confirmed as having the virus based on RT-qPCR did develop antibodies within that time frame. And then a number of them also developed the neutralizing antibodies. So that's kind of another key point to understand as we hear a lot about that these antibody tests aren't necessarily directly demonstrating immunity. It is thought that one of the key aspects of antibody-mediated immunity excuse me, is the generation of neutralizing antibodies. So these are those antibodies that directly bind to the spike protein and prevent the ability of the virus to infect the cell. So that's definitely an important thing to understand. That type of testing, though, is very difficult to do for many reasons, but one of them is that it typically requires a high-level biosafety lab, so it makes that sort of test much more difficult and challenging to do. So it's not something that's at least yet something that can be set up for widespread implementation. And they go on to say declining neutralizing antibody titers were observed during the follow-up period. While some individuals at high peak, so this is based on their measurements, so greater than 10,000, maintained titers greater than 1,000 at more than 60 days post-symptom onset. Some with a lower peak had titers approaching baseline within the follow-up period. So the general idea is that the higher the titers, the longer they may be maintained. The lower the titers, then they may tend to decline below baseline faster. They go on to say, we suggest that this transient neutralizing antibody response is a feature shared by both the SARS-CoV-2 infection that causes low disease severity and the circulating seasonal coronaviruses that are associated with common colds. So again, this generated a lot of concern, especially based on media reports that this supposedly demonstrated that neutralizing antibody responses only lasted a certain period of time. It still doesn't really fully address that issue for a number of reasons. I mean, it is known that by and large, during acute infection and even sometime after that, antibody levels peak and then they tend to decline. So to some extent, that's sort of an expected phenomenon. But what they don't really know is if some of these levels, antibodies still persist long-term, possibly at low levels that are too low to be detected in some of the tests that are out there, and whether that still provides significant protection. So that's still an open question as to whether lower levels of antibodies can still provide significant protection. And again, based on previous studies of other viruses, there are cases where that definitely does seem to be the case, where these neutralizing antibodies at low levels can still confer protection. So this is some of the data from this particular study. This particular figure, and you can see the title here at the bottom, Kinetics of Antibody Development Against SARS-CoV-2 Antigens Over Time. So they're looking at antibodies to the receptor binding domain of the spike protein. That's the RBD on the left, and then the nucleocapsid protein, that's on the right. And then they're looking on average at the development of seropositivity, meaning the time point at which antibodies were detected in different individuals. So again, this is for IgG results, and then on the right y-axis it says percent seropositive. So that's basically just telling you whether or not patients were positive for antibodies. And then the x-axis is days post-symptom onset. So for both of these graphs, you can see that on average, again, right around 2 to 3 weeks is when the majority of people have achieved antibody responses. This is really confirming what we know from many other studies as well. This is actually illustrating the individual differences or variation within those responses. So those curves look nice and clean, but when you look at the individual data, it's much more scattered. So this is looking specifically at the receptor binding domain antibodies, and then it breaks it out by IgM and then IgG, and that's on the top row. And again, the x-axis is days after symptom onset. And then the color coding for the various dots is indicated here on the right. So severity score from very low or asymptomatic all the way to very severe cases. So what you can see here is even though there's sort of these general curves that they can generate from the data, there's still a lot of scatter. So that's one of the key take-homes here is when you're looking at antibody testing, not every patient is going to follow the classic pattern. This is just looking a little bit more clearly at the differences between the lower severity or mild groups versus the high severity groups for the different antibodies. So this is IgG on the left, and this is the basically the antibody levels indicated. In this case, it's for the spike protein. So you can see here for low severity level on the x-axis, 0 to 3, there's a lot of scatter, but on average, so they indicated the average by the bar here, it's somewhat lower than patients who are have much more severe symptoms. For IgM, there's also that same dichotomy. So once again, you can see there's some folks in the low category that had relatively mild cases that still had fairly high antibody titers. And then there's some folks in the high severity group that had lower antibody titers, but on average, they tend to have higher levels. So once again, just note that there is some significant individual variability. This is kind of the key observation that drew a lot of attention in terms of overall trend towards decline in neutralizing antibodies. So this is a chart looking at neutralizing antibodies across all these patients, again by severity score. And then on average, those neutralizing antibodies tended to decrease during this time period by about up to 100 days. Although you can see based on the scatter that some patients still had fairly high levels at that point. So really beyond that is kind of the open question is, We do expect them to decline to some extent, but does that decline amount to lack of immunity at some point, or is there some persistent immunity? Okay, so that's a lot of data that I went through here. I just want to summarize some of the key points from these various studies. So among the key observations is that most patients produce detectable antibodies within the 2 to 3 week time frame. So from that point on, up to a certain point, that's really the time window where patients are most likely to be positive for antibody testing. And again, that translates to sensitivity. So the sensitivity will likely be highest for antibody tests during that time period. IgM antibodies in general decline sooner than IgG antibodies, typically within 4 to 6 weeks. Again, there's variability. IgG antibodies may decline over time as well. Those are usually the longer term antibodies. So again, we don't have enough time yet under our belt to really look at that long term, but hopefully that gets clarified so that we better understand that in the short term. Unclear if low level neutralizing antibodies persist long term. So that's really the main thing that research is focused on, is trying to understand, does this long-term immunity persist, and how relevant is it? Okay, so this next study here is titled Seroprevalence of Antibodies to SARS-CoV-2 in 10 sites in the United States. This is looking at data from fairly early on in the pandemic, March 23rd to May 12th, although it was recently published in the Journal of American Medical Association. So this is one of the most comprehensive studies published to date and peer-reviewed, looking at the seroprevalence data. And this is, again, from several sites in the US. I'm just going to go ahead and read through the whole results section from their summary. Serum samples were tested from 16,025 people, over 8,000 of whom were women, 1,200 were 18 years and younger, 5,800 were 65 and older. Most specimens from each site had no evidence of antibodies to SARS-CoV-2. Adjusted estimates of the proportion of people seroreactive to SARS-CoV-2 spike protein antibodies range from 1% in the Bay Area to 6.9% in the New York City area, collected March 23rd to April 1st. So again, very early on in the pandemic. But the key take home, and this is again something that was really emphasized quite a bit in news reports recently, is that the estimated number of infections range from 6 to 24 times the number of reported cases. And then for 7 of those sites, an estimated greater than 10 times more SARS-CoV-2 infections occurred than the number of reported cases. So this is really illustrating one of the key important benefits of doing antibody testing, to look at it kind of in a more epidemiological setting, where we're getting an idea of the overall seroprevalence in the population. And this is fairly consistent from other studies that have shown that overall the estimates of actual infection tend to be at least 10 times higher than the known cases. All right, so that's mostly concluding the antibody research section. So I just want to kind of wrap up here and talk a little bit more about overall immunity, and focusing on some newer research that's coming out about T-cell immunity and how that may be important as well. So the study was titled SARS-CoV-2 specific T-cell immunity in cases of COVID-19 and SARS and uninfected controls. We showed that SARS recovered patients still possess long-lasting memory T cells reactive to SARS NP, or nucleocapsid protein, 17 years after the 2003 outbreak, which displayed robust cross-reactivity to SARS-CoV-2. Surprisingly, also frequently detected SARS-CoV-2 specific T cells in individuals with no history of SARS, COVID-19, or contact with SARS-CoV-19 patients. Thus, infection with beta coronaviruses induces multi-specific and long-lasting T-cell immunity to the structural protein NP. So this is one of the first studies that was looking at another aspect of long-term immunity. So of course, on the one hand, we have memory B cells and long-lived plasma cells that are generating antibodies. Then we also have cell-mediated immunity based on T-cell immunity. And it does appear that that can develop and then be specific to SARS-CoV-2, partly in response to past reactivity to other coronaviruses. They go on to say, understanding how pre-existing NP and then ORF1 is another antigen from the virus. So these two antigen-specific T cells present in the general population impact susceptibility, and pathogenesis of SARS-CoV-2 infection is of paramount importance for the management of current COVID-19 pandemic. So there's a couple of studies that have been published recently. Actually, these are not quite peer-reviewed yet, so they're on the preprint server by our XIV. So one is titled Broad and Strong Memory CD4 and CD8 T Cells Induced by SARS-CoV-2 in UK Convalescent COVID-19 Patients. And then the second one is Robust T Cell Immunity in Convalescent Individuals with Asymptomatic or Mild COVID-19. So really the bottom line from all this is there's growing evidence that most individuals who have SARS-CoV-2 do develop antibody responses. In general, it does appear that most people do produce neutralizing responses based on studies to date. It's unclear whether or not those neutralizing antibodies last long term. And then we do know that they probably decline based on some newer studies that have been coming out, but we still don't know whether those lower levels may still be protective. And then in addition, we also know that T cell immunity does appear to be particularly important long-term as well for immunity to SARS-CoV-2. So with that, hopefully that helps bring you up to date on some of the new research that's been coming out, particularly with regard to the timeline of development of antibodies, and then also the concept that people who have more severe symptoms tend to have higher levels than individuals who have mild cases or even asymptomatic cases. So that's important information to take into account when you're looking at antibody tests in relation to the particular symptom in their particular patient and their symptoms. So thank you so much for listening. We do have a lot more information on our website about our testing, as well as a number of learning resources at Diagnostic Solutions Lab. So thanks again.